Hey everyone, and welcome to The Deep Dive. I always try to cater these deep dives to our audience, and I know we've got a lot of medical microbiologists listening in.
Always exciting to talk to a specialized crowd.
Right, so today we're going to really get down to the nitty-gritty with a fascinating infection.
Oh, yeah.
Donovanosis.
Ah, Donovanosis. Now, that's one we don't see every day.
Exactly, and that's what makes it so interesting, right? Even its name is a bit of a mystery, like, should we call it Donovanosis or granuloma inguinale?
Well, both names have been used, but I think most people prefer Donovanosis these days. It's just more specific.
Yeah, it rolls off the tongue a bit better, too. Plus, there's this whole debate about what to call the bacteria itself.
Oh, right. The Calymmatobacterium granulomatis versus Klebsiella granulomatis conundrum.
You got it. So much to unpack. Where do you think we should even begin?
Well, I guess we should start with the basics. What is Donovanosis anyway? Like, what's going on at a microscopic level?
Great point. So it's a bacterial infection, and the real giveaway when you're looking under the microscope is something called a Donovan body.
Ah, yes, those classic Donovan bodies.
Right. These little intracellular inclusions tell you pretty much all you need to know.
Well, they're more than just inclusions. They're actually clusters of bacteria living inside macrophages.
Whoa, inside.
Yep, intracellular. And that's a big part of why this infection is so unique.
So that's what contributes to those ulcers, huh?
Exactly, those characteristic beefy-red ulcers.
Yeah, I've seen pictures. I won't soon forget those.
Well, hopefully you won't see them too often in real life. But speaking of things we can see, let's shift gears a bit and talk about the culprit itself, the bacteria.
Calymmatobacterium granulomatis.
What was originally called that, yeah.
It's a bit of a mouthful, isn't it?
Yeah, it's not exactly the most elegant name, and as you mentioned earlier, that's where the debate comes in. Some researchers think it should be reclassified as Klebsiella granulomatis.
And they've got a pretty good case, right?
Yeah, in recent decades, we've finally been able to culture this bacterium, which opened the door for genetic analysis. Turns out it's extremely closely related to the Klebsiella species.
Like, how close are we talking?
Almost 99% homology in a key ribosomal RNA gene. So it's really a question of how much weight we give to genetic relatedness versus clinical presentation.
So it's like that distant cousin who looks and acts just like everyone else at the family reunion, but insists they're totally different.
Exactly. Some researchers are firmly in it's a Klebsiella camp, while others are holding on to the original classification.
So how do we even begin to tease this apart? Like, what makes Calymmatobacterium granulomatis or Klebsiella granulomatis so unique?
Well, a lot of it comes down to its lifestyle. You see, unlike many other bacteria, this one prefers to live inside cells.
Inside cells.
Yep, specifically histiocytes. It's an intracellular dweller.
So it's like it has its own little hideaway?
Precisely, and that has a lot to do with those distinctive ulcers we see with Donovanosis.
Makes sense. Something else I found fascinating is that this bacterium seems to be missing a key gene.
Oh, you're talking about the SCORA gene?
Yes. The one responsible for transporting sugars.
Right, and researchers think that might be why it's forced to live inside cells where it can access nutrients more easily.
Fascinating. So it's like it traded its sweet tooth for a cozy intracellular apartment.
I like that analogy. And you're right, that does have implications for treatment, but we'll get to that later.
Sounds good. First, though, I'm curious about where this bacterium likes to hang out geographically. Does it have like a favorite continent or something?
Well, more like endemic regions. It's mostly found in tropical and subtropical climates, places like India, Brazil, Papua New Guinea, parts of South Africa.
So not everywhere.
Right. Interestingly, it's been virtually eradicated in Australia, and it's pretty much disappeared from the southern U.S.
Really?
Yeah. Targeted eradication campaigns have proven to be quite effective.
Wow, that's encouraging. So how does this bacteria actually get around? How is it transmitted?
Primarily through sexual contact. However, there have been rare cases of mother-to-child transmission during delivery.
Okay, so that's something to keep in mind. And speaking of keeping things in mind, how common is Donovanosis anyway? I feel like it's not something we hear about very often.
You're right. It's not as common as some other STIs, but its prevalence varies significantly depending on the region. For example, in certain areas of India and Papua New Guinea, it's responsible for a significant proportion of genital ulcer diseases.
Wow, that's much higher than I would have guessed.
It just goes to show that even though we might not see it every day, it's important to be aware of this infection.
Absolutely. So we've got this unique bacteria, Calymmatobacterium granulomatis, causing these distinctive ulcers. And lurking in specific parts of the world, what happens next? Like, how does this infection actually progress in a patient?
Well, that's where things get really interesting. The clinical presentation of Donovanosis can be quite varied, and it's not always as straightforward as you might think.
I'd love a bit of clinical intrigue. Let's dive into that.
Okay, so the classic presentation, it usually starts with a single or multiple ulcers.
Okay.
Usually in the genital area.
Yeah, that's what I was thinking of too.
Painless too.
Oh, painless. Interesting.
Yep. But as I said, it can get a lot more complex.
More complex? How so?
Well, these ulcers can spread.
It's great.
Yeah, and the infection can even progress to the lymph nodes.
So it's not just a local infection then. It can actually become systemic.
Yeah, in some cases, the bacteria can travel to the lymph nodes, causing swelling and sometimes even abscesses.
Oh, wow.
And in very rare cases, it can spread through the bloodstream to other parts of the body.
So we're talking like a really serious infection, potentially.
It can be, yeah. If left untreated, Donovanosis can lead to some pretty nasty complications, scarring, lymphatic obstruction,
Oh, yeah.
and even an increased risk of certain types of cancer.
Really? Cancer, too.
Yeah, there was this fascinating case in Australia, I think it was, where a patient presented with a whole range of symptoms. Abscesses in different locations, oral lesions, even spinal cord compression.
Wow. That's a lot.
Yeah, it really highlights the importance of early diagnosis and treatment.
Absolutely. But that brings us to another challenge, right? Accurately diagnosing Donovanosis.
Right, because those ulcers can look a lot like other conditions.
Exactly. So how do we tell Donovanosis apart from something like syphilis or chancroid?
Well, that's where those Donovan bodies we talked about earlier come into play.
Ah,
Microscopic examination of a sample from the ulcer is the gold standard for diagnosis.
so we're literally looking for those clusters of bacteria inside macrophages under the microscope.
Yep, exactly. You usually stain them with special dyes, like Giemsa or Wright's stain to make them easier to see.
Right.
But even then, you need to have a trained eye because sometimes these Donovan bodies can be mistaken for similar inclusions that are found in other diseases.
Like what?
Leishmaniasis, for example.
Ah, okay. So it really does take experience and a good microscope.
It does. Are there any other diagnostic tools available?
Well, there are some newer tools like PCR and serological tests, but they're not widely available yet.
Yeah, I've heard about those.
Mostly used in research settings. For now, microscopy is still the most reliable way to confirm a Donovanosis diagnosis.
Makes sense.
Oh.
So let's say we've got a confirmed case of Donovanosis. What's next? How do we actually treat this tricky intracellular bacteria?
Well, the good news is it is treatable
Ew.
with antibiotics.
Okay.
Azithromycin is the current first-line treatment of choice.
Azithromycin. Why azithromycin?
Well, it seems to be particularly effective against these intracellular bacteria, and it's generally well tolerated. But to be honest, head-to-head clinical trials comparing different antibiotics are lacking, so more research is definitely needed in this area.
Okay, so what are the other options if azithromycin doesn't work or if it's not available?
Well, doxycycline, erythromycin,
Okay.
and some of the fluoroquinolones have also been shown to be effective.
I see.
The key is to continue treatment until the lesions are completely healed, which usually takes a few weeks.
So patience is key.
Absolutely.
Yeah.
And sometimes a change in antibiotics might be necessary,
Oh, okay.
or even surgery in cases with really severe complications.
That makes sense. But it's not just about treating the individual right. We need to think bigger picture here and consider how to prevent the spread of this thing.
Exactly. And that brings us to partner management and public health measures.
Right.
So let's break those down.
Yeah.
What does effective partner management involve?
Well, it's all about identifying and treating the sexual partners of someone who's been diagnosed with Donovanosis,
Got it.
even if they don't have any symptoms.
So kind of like contact tracing.
Exactly. It helps to break the chain of transmission and prevent reinfection. And that's where public health programs and education campaigns come in.
Yeah, I can see how that would be important.
Raising awareness about Donovanosis, its symptoms, the importance of seeking treatment, all of that can go a long way in reducing the spread.
Right. But I imagine stigma can be a huge barrier.
Oh, absolutely. Addressing the stigma surrounding STIs is so important for any successful prevention and control efforts.
Yeah, that makes sense. I've talked a lot about the challenges of Donovanosis, the varied clinical presentations, the difficulty in diagnosing it, the importance of preventing its spread. But there's also a positive side to this story, right?
Absolutely. There are some valuable lessons to be learned from the successes of eradication campaigns like the one in Australia.
Yes. The Australian eradication campaign. It's like the gold standard, isn't it?
It's pretty impressive. What made it so successful? It's pretty impressive.
Yeah.
What made it so successful?
Yeah, what was their secret?
Well, it wasn't really a secret, more like a combination of factors.
Okay, like what?
They really engaged the community. They educated both the public and healthcare workers and made sure that diagnostics and treatment were readily available.
Smart.
Plus, they implemented this really robust surveillance program.
Oh, yeah, to track cases and monitor progress.
Exactly. So it was a multi-pronged approach, targeting the disease from all angles.
And it worked.
It did. They were able to virtually eliminate Donovanosis from Australia.
Wow, that's incredible.
But as we've discussed, other endemic regions face some pretty unique challenges. So could this model really be replicated elsewhere?
It's a tough question. The Australian campaign definitely provides some valuable insights, but there's no one-size-fits-all solution.
Right.
Resource limitations, cultural barriers, even political complexities can all play a role.
So it's really about adapting those core principles to the specific context of each region.
Exactly. Taking into account the unique challenges and opportunities.
And that's where medical microbiologists like you come in.
Oh, yeah.
You have such a deep understanding of the unique challenges and opportunities within your own community.
It's true. We're kind of like the boots on the ground.
Exactly. Speaking of boots on the ground, tell me, have you ever had any cases where the diagnosis wasn't immediately clear?
I'd say that's more common than you might think.
Really?
Yeah, sometimes those ulcers can be tricky, They can mimic other conditions.
Yeah, that's what we were talking about earlier.
Exactly. It's important to keep a broad differential diagnosis in mind, for sure. So any memorable cases come to mind?
Let me think. Oh, yeah. A few years back, I had this patient who came in with what looked like a typical Donovanosis ulcer. But something about the location and the patient's history just didn't quite fit.
Interesting. What did you do?
Well, I decided to order a biopsy just to be safe.
Good call.
And it turned out to be cutaneous leishmaniasis.
Wow. So it can really look like Donovanosis.
It can. Yeah, it was a good reminder to never jump to conclusions based solely on clinical presentation.
Absolutely. Always got to confirm with those lab tests. Speaking of lab tests, what are your thoughts on the newer diagnostic tools like PCR and serological tests? Do you think they'll eventually replace microscopy as the gold standard?
It's hard to say. And PCR in particular offers the potential for much greater sensitivity and specificity.
Right.
But right now, access is limited, and we need more standardization before it can really become the go-to method.
Makes sense.
Yeah.
It's definitely an area ripe for further research and development. Now, switching gears to treatment, we talked about azithromycin as the preferred choice. But what about those alternative antibiotics? Have you had much experience with those?
Yeah, I've used doxycycline in a few cases where azithromycin wasn't an option, either because of allergies or availability issues.
Yeah.
And it's generally been pretty effective.
That's good to know.
Yeah.
Always good to have backup options. Okay, so let's zoom out for a minute and revisit that big picture question. Could the Australian eradication campaign really be a blueprint for the rest of the world?
It's a complex issue. Like we've discussed, there are so many factors to consider.
Right.
A cookie-cutter approach just won't work everywhere.
Of course not.
But I do believe that the core principles of that campaign, community engagement, education, access to care, and robust surveillance, those are all universally applicable.
Yeah, I agree. It's about taking those principles and adapting them to the specific context of each region.
Exactly. And that's where I think medical microbiologists have such a crucial role to play.
Absolutely.
Yeah.
Well, this has been an incredibly insightful conversation. I've learned so much about Donovanosis.
It's been my pleasure.
Any final words of wisdom for our listeners, especially those who might encounter Donovanosis in their practice?
I would say stay curious, keep learning,
Great advice.
and never underestimate the power of a good microscope.
So true. And to our listeners out there, thank you so much for joining us on this deep dive into the fascinating world of Donovanosis. We hope you've gained some valuable knowledge that will help you in your practice and beyond. Until next time, stay curious, keep exploring, and remember, even the smallest microbes can teach us profound lessons.